Okay, last week we discussed about Molly's test. I told you that this test is based on the hydrolyzing and dehydration action of sulfuric acid on carbohydrates, and this test can be used to identify any carbohydrates. Okay, do you have any questions to clarify based on Molly's test? Okay, if not, I will move on to the next test that we are going to discuss with you today. That is Morse test. Okay, even in the last week, I told you that Morse test is not a well established or well used method in biochemistry experiments, because the principle behind this is not a very solid one. Okay, that is when we add in concentrated bases or alkaline to reducing sugars, that will form enolic substances. Okay, and these enolic substances are quite unstable. Therefore, okay, then one has a question that do Molly's test gives positive results for trioses and tetroses? Okay, what can I say for that? It will give positive results, but the observations that we expect for Molly's test for hexoses, pentoses, and other disaccharides and polysaccharides, I mean carbohydrates, that would be different than the results that will give for trioses and tetroses. And the primary objective of this method is established to identify actually monosaccharides such as pentoses and hexoses, as well as disaccharides and other polysaccharides, not specifically for trioses and tetroses. Okay, even in this biochemistry, we didn't give much concentration on trioses and tetroses. Even I think even in your theory classes. Okay, so I think that's clear. That can form colored compounds with alpha naphthol, but I'm not sure that the result is much similar to that of a purple coloring that will give for hexoses and pentoses. Okay, for an example, the glucoses, fructoses, galactoses, like sugars. Okay, the result may be little bit different than that of hexoses and pentoses. Okay, so Morse test. So when we are okay, first of all, tell me what is meant by a reducing sugar. What does that mean? Reducing sugar, reducing sugars, non-reducing sugars. I think you have heard about that by now. Oh, before moving on to the reducing sugars, what is meant by a reducing agent and oxidizing agent? That is basic chemistry. What is that? Reducing agent or reductant or reducer or electron donor. We can give many names. Reducing agent. What is that? Reducing oxidation number. Okay, that is true. If you define it in a bit advanced manner, what can you say? The substance which cause oxidation. Yes, good. Anuttara, yes, means the substance which cause for oxidation. Okay, actually, you know that in every redox reaction, there is a electron transfer. Okay, one will give electrons and one will receive electrons. Okay, then reducing agent or reductant or reducer or electron donor is the agent or any molecular substance or any substance which will donate electrons. Okay, and the oxidizing agent will receive electron, and that is called electron recipient. Okay, and we can define reducing agents as substances which lose electrons to other substances in a redox reaction and gets oxidized to higher state. Okay, and reducing agents will oxidize. Okay, and the oxidizing agents will reduce. Okay, don't confuse that. Before we talking about reducing sugars and non-reducing sugars and other things, you need to know the principle. Okay, what is meant by a reducing agent? Okay, is that clear? Reducing agent එකක් කියලා කියන්නේ redox reaction එකකදී electron donor electron ලබා දෙන කෙනෙක්. එතකොට reducing redox reaction එකකදී reducing agent වෙන්නේ ඇත්තටම oxidize වෙන්නේ. Oxidizing agent එක තමයි reduce වෙන්නේ. Reducing agent, we give that name because that substance can reduce other substances. Okay, and that particular substance will oxidize. Okay, can you give examples for reducing agents? Reducing examples for reducing agents. Who can give electrons? Yes, Vatsala mentioned that sulfur dioxide H2 is not a good example for reducing agent, I think. But sulfide compounds, formic acid, and earth metals are good examples for reducing agents. Okay, then if we move on to what we intend to talk, reducing sugars, then what is meant by a reducing sugar? And what functional group act as a reducing agent? In particularly in monosaccharides, you have learned about the structure of monosaccharide. So why we call some monosaccharides, almost all monosaccharides are reducing agents? So why is that? What could be the reason for that? Basically, what is meant by a reducing sugar? Then why we call all monosaccharides are reducing agents? What is the reason for that? And why some disaccharides cannot act as reducing sugars or reducing agents? What is that? What is the principle behind that? Always know the principle or the theory behind the things. Why is that? Okay, I'm not talking about theoretical things, but in order to understand these experiments, you need to know you need to understand the theory. Okay, that's why I'm talking. Actually, reducing sugars are those which behave in a similar as that of reducing agents because of the free ketone or aldehyde group. Okay, you know that if we tell that in a simple manner, reducing sugars are the sugars that behave as a reducing agent which have free aldehyde or free ketone groups. You know that we use to denote free aldehyde and free ketone groups as hemiacetal functional groups. Okay, then those sugars which have free aldehyde and free ketone groups are called reducing sugars. Okay, then non-reducing sugars, that is quite obvious that they do not have free aldehyde or ketone groups. Okay, then tell me, what are the examples for non-reducing sugars? You know that in the qualitative analysis of carbohydrates, then in most tests that we're going to discuss with you are to distinguish reducing sugars and non-reducing sugars. Okay, in some cases in biochemistry experiments we do need that. Okay, then tell me, what are the examples you know for non-reducing sugars? It means they do not have free aldehyde and ketone groups to donor electrons or to give electrons. Okay, and they basically they cannot oxidize. They cannot further oxidize. Okay, oxyකරණය වෙන්න බෑ ඒගොල්ලන්ට තවදුරටත්. Sucrose and trehalose. Okay, yes, good. Sucrose and trehalose. Yes, Vasala. Yes, you're correct. Sucrose, trehalose, raffinose, stachyose are good examples for non-reducing sugars. Okay, this most tests can use to identify reducing sugars. Okay, because reducing sugars can react with concentrated bases or alkaline substances, and they will form enolic substances. That process is called enolization. Okay, in the enolization process, the resulting substances are very unstable. Thereby, if we add acids to that, then the reaction will be reversible. Okay, in here we are going to take we have to take carbohydrate into a test tube, and we have to add 20% sodium hydroxide and heat. And you will observe when we doing that, reducing sugars will give yellowish color solution. And when we acidify the solution with sulfuric acid, you will observe that this color change will be reversed. Okay, so I will show the video and observe that carefully.
Today your first practical is Moore's test. We are going to do this practical for the carbohydrates. Here you can see we have selected 5 carbohydrate solutions. First one is glucose, fructose, lactose, maltose and sucrose. First of all, you have to get 10 milliliters of each solution for the test tube. Before that, you have to take these solutions into a beaker. So we are going to get solutions from each sugar solution from the beaker. Okay, now we are going to get 10 milliliters of each solution to a test tube. Okay, then as the next step, you are going to add 5 milliliters of 20% sodium hydroxide into all these solutions. Here we are going to use a dropper to add sodium hydroxide. Then you have to boil this in a Bunsen burner. In boiling, make sure that you are not going to boil it for a continuous period. Otherwise, it will make some explosions. And while boiling, you have to observe the color change. Now you can see the color is changed from colorless to yellowish color. Now you can see this color has changed from the colorless to yellowish color. Now we are going to let it cool for few minutes before adding sulfuric. Why we are going to let it cool? Because this solution is already boiled, and if we are going to add sulfuric in that moment, it will cause some explosion. Therefore, we have to let it down cool for some time. Next, you are going to add corn sulfuric to the sugar solution. When you add sulfuric, you make sure that you are going to add it along through the walls of the boiling tube. Okay? And make sure that you are going to add this in drop wise, not in a flow. And observe the color change. Gradually, the color of the solution is going to fade and it will become colorless. So now you can see the yellow color of the solution has become colorless after adding concentrated sulfuric. Now we are going to see what will happen if we add more sulfuric to the solution. Okay, now you can see what will happen when we are adding more sulfuric to the solution. In here the solution has caramelized. You can see that there's a brown color patch or something in the bottom of the boiling tube due to the caramelization of the sugar. We are going to do the same procedure for the other sugar solutions. Now I am going to add 5 milliliter of 20% sodium hydroxide in the lactose solution. After adding sodium hydroxide, you have to repeat the same procedure as I described before. Now you are going to boil the sugar solution and observe the color change. Now you can see the color change. The color has changed into yellowish color from the colorless. Okay. Now we are going to let it down cool. Next you are going to add 5 milliliters of 20% sodium hydroxide into the maltose solution. And boil. Now you can see the color change from the colorless to the yellowish color. We are going to add same amount of sodium hydroxide into the fructose solution. And boil in the Bunsen burner and observe the color change. See, in here also the colorless solution has become into the yellowish color. To add same amount of sodium hydroxide into the sucrose solution. And boil in the Bunsen burner and observe the color change. You can see the color of the sucrose solution has not changed. It was already colorless and now also it is colorless. Now you can see the color changes of the sugar solutions after adding 20% sodium hydroxide. Here is the sample that we had. So corn sulfuric, it has already caramelized, and here is the lactose solution and maltose solution, fructose, and here is the sucrose solution.
Then, as you observed, we used 5 sugars, glucose, maltose, lactose, fructose, and sucrose. And among those 5 sugars we used, 4 sugars gave the positive results, and sucrose didn't give the positive result. Because you can see, if you compare the difference between sucrose and other sugars we used, you know that sucrose is a non-reducing sugar. Okay, this is the process I told you about enolization. Then, and here this is a free aldehyde group. You know that in reducing sugars there's a either free aldehyde group or ketone group. So when there's sodium hydroxide, concentrated sodium hydroxide, and heat, this will form enolic substances. Enol means enol, hydroxyl group with double bond. Okay, these enolic substances are yellow in color and they are quite unstable. Therefore, when we add sodium hydroxide, it will form enolic substances which is yellow in color. And when we add acid, concentrated acid, that process will be reversed. Okay, that is why when we add in concentrated sulfuric acid, the yellow color disappeared. Okay, so this is the principle. And I want to tell you that even though I discussed this with you, this is not a good qualitative experiment to distinguish reducing sugars. Okay, even you search this in good biochemistry books or even in the internet, you will not find much information on most tests. Okay, there's a limitation of this test. Okay, is that limitation of using most tests to distinguish reducing sugars is that when we add concentrated base and when we heat in that in an open flame, you know that there's a process called caramelization. Okay, every sugar can be caramelized. Every sugar can be caramelized. What is caramelization? What is meant by caramelization? Every sugar has its own a temperature to be caramelized. For an example, for glucose, it's about 105 degrees Celsius, and for sucrose, it's about 180 degrees Celsius. Okay, and if we talk about caramelization, caramelization is a non-enzymatic browning reaction, and it is poorly understood process yet, but in this chemical process, it will form many intermediate chemical products. Okay, that chemical products has flavor and aroma. Okay, that is why we do use caramelization in cooking. Okay, in the caramelization process, diacetyl, caramellins, caramellans, like many products can be formed, and these products have a unique flavor as well as aroma. Okay, so caramelization is accelerated by concentrated bases as well as concentrated acids. Okay, that's why when we add in high amounts of sulfuric, that you have observed that that has caramelized, as well as if we boil the content for a longer period of time, that will also caramelize. Even for sucrose, if we expose sucrose to 180 degrees Celsius, and it is a quite higher temperature that we have used a open flame, and if we keep that for another 5 minutes, that definitely sucrose will also caramelize. And you know that Caramelized has a residuous color, usually yellow color. So using this Morse test, we cannot, it is not a good test to distinguish reducing sugars. Okay, did you get my point? පැහැදිලිද මේ Morse test එක කියන එක වැඩිපුර පාවිච්චි කරන test එකක් නෙමෙයි. So I talk this with you because this is in the curriculum, but this is not used in the real world. Okay, because the expected results can be interfered with the caramelization process. Okay, we have very good other simple biochemical experiments to distinguish reducing sugars. Okay, we will discuss in this session also. Therefore, if you are given to distinguish reducing sugars and non-reducing sugars, then never use Morse test. That is my advice. Okay, even in the biochemistry practical exams, we will give few sugars with the same appearance, same flavor, same order. Then you have to distinguish. You have to sometimes identify whether this is a reducing or non-reducing sugar. So you have a range of tests you studied and procedures in the examination. So you have to use a good test and identify and give the results, come up with results. So due to the limitations of this test, this is not widely used. Okay, so we have other good tests to distinguish reducing sugars and non-reducing sugars. What are the tests you know? Other tests in advanced level chemistry, you have learned about those tests. Two major tests. What is that? Benedict test, Anitya Fehling's test. Okay, in this under biochemistry experiments, we will talk about Fehling's test. Okay, first of all, let me ask you, have you all studied chemistry in your advanced level? Are there anyone who didn't follow chemistry? That's okay. There's no rocket science in this subject, but I just want to know. So this is a few extension of advanced level chemistry. Okay, there's no much deep chemical things. Okay, just to refresh your knowledge. Okay. So as the third experiment, I will discuss with you about Fehling's test. Okay. Then Fehling's test is used to identify reducing sugars. All monosaccharides and most of the disaccharides are reducing sugars, and they have the capacity to reduce alkaline solutions of copper sulfate, silver, mercury, bismuth, acid solutions of molybdate and selenium acid, organic compounds such as Picric and Methylene blue. So these are mild oxidizing agents. Okay, then as well, Fehling solution as well as Benedict solutions are mild oxidizing agents. Okay, that is the principle of this test. Okay, reducing sugars can react with mild oxidizing agents. Okay, mild oxidizing agents. You know that reducing sugars, they are reducing agents, thereby they can do redox reaction with oxidizing agents, especially with mild oxidizing agents. Okay, and you know that Fehling solution consists of two components, one Fehling's A and Fehling's B. Okay, which one of these two components is blue in color? Which one of among the two components in Fehling solution, Fehling's A and B, which one is the blue? Yes, miss. Fehling's A. Failings A. Failings is A contain copper sulfate and failings B contain Rochelle salt and sodium hydroxide. You know that copper sulfate is in aqueous medium, it is blue in color. Okay, then in order to prepare failings A and B, we have to mix the similar amount of failings A and B and give add doubled the content with distilled water. Okay, and what is the color of failing solution? There's a specific name for that. What is the color of failing solution? Is it
Blue.
Yes, blue. Specifically Persian blue. Okay, when we add failings A and failings B and prepare the failing solution, that is also blue. We have a specific name for that, Persian blue. Okay, then what is Rochelle salt? It is what is that? It is sodium potassium tartarate. Okay, you know that, you know these things. Okay. Okay, then first of all, I will discuss with you how to prepare failing solution. Okay, when we prepare in the failing solution, you have to mix similar amounts of failings A and B. Say, according to this procedure, mix 1 milliliter of each failings A and B solution in a test tube and add 2 milliliters of distilled water and boil the mixture. Okay, then what can you tell you about the acidity of this failing solution? Should it be acidic or slightly basic? What can you say about that? Should it be acidic or slightly basic? After preparing failing solution, definitely you have to check that with litmus. Okay, acidic or basic. Then this contains sodium hydroxide. Usually, the failing solution should be slightly basic, and we always, when we need failing solution, we prepare that freshly. Okay, we do not store failing solution for a long period of time. Okay, then let me ask, what is the active component of failing solution? What is that? What is the active component in the failing solution? You know these things, ligands and the things. What is that? We start to cuprate 2. Yes, Cu2 plus. We can say that Cu2 plus, and the actual complex is, you know that copper is a D block element, and this can form ligands. The complexes with ligands, and the complex is bitartrate to cuprate 2. Okay. Then in the failing test, yes, the final failing solution should be slightly basic. Therefore, we have to add sodium hydroxide if necessary and check with the litmus. Okay. Then what will happen if we add red litmus paper to failing solution? Will it be remained red, or will it turn into blue? Red litmus. If it is basic, what will happen? Ratu litmus කඩදාසිය නිල් පාට වෙනවද නැත්නම් රතු පාට වෙලාම තියෙනවද? What can you say?
Red litmus turn into blue color.
Yes. You are very, as you mentioned very correctly, red litmus paper will turn into blue if it is slightly basic. And if we do not have red litmus in our laboratory, we can use blue litmus. Then blue litmus remained blue. Okay. Then when we doing the this failings experiment test to distinguish reducing sugars and non-reducing sugars, we have to take 2 milliliters of 1% sugar solution, or the sugar solution given, into a test tube and add 2 milliliters of failing solution, and we have to boil the mixture. Okay, then you will observe a color change. Then what is the final observation? What will be the final observation in failings test? You know that. Okay, you will observe a series of color change. Okay, first blue color, then green, yellow, orange, and finally you will observe a brick red precipitate if the sugar solution contain reducing sugars. Okay, that is uh, the principle of this test. We will talk about that a little bit deeper. You know that this is the advanced level knowledge you have. So we will talk about that little bit deeper. Before that, I will show the practical video or procedure that we have recorded in our laboratory.
Okay, now we are going to do another qualitative practicals for the carbohydrates. It is Fehling test. For that we need Fehling A, which is have blue color. As you can see, it is a blue color solution. And then we need Fehling B. It is a colorless solution. And we need distilled water. To prepare Fehling solution, we need 1 milliliter of the Fehling A, 1 milliliter of the Fehling B, and 2 milliliters of the distilled water. Now I am going to make Fehling solution for the all 5 carbohydrates: sucrose, maltose, lactose, fructose, and glucose. For that, I am going to measure 5 milliliter of the Fehling A. Milliliters of the Fehling B. And we are going to mix it together. As you can see, it turns into Persian blue color. Then we are going to add 10 milliliters of the distilled water. This is our Fehling solution. Now we are going to boil it by using Bunsen burner. We don't need to boil it for a long period of time. We boil it 1 to 2 minutes to remove the dissolved oxygen in the solution. Okay, now we need red litmus, we are going to check the saline solution with red litmus. When we put red litmus, it turns into the blue color. So the required amount of acidity already there, so we don't need to add sodium hydroxide. Then we need sugar solution. We need 2 milliliter of each sugar solution, and we are going to add 2 milliliters of the Fehling. Now we are going to measure 2 milliliters of the each sugar solution. Then we have 2 milliliters of saline solution. Okay, then we are going to put it in the water bath. Before that, before put it in the water bath, you can see fructose start to react with saline solution. As you can see, the blue color turns into the mud green color because fructose has higher reaction rate when compared with the other sugar solution. Now we are going to put this sugar solution into the water bath. Then we can observe the color change. As you can see, fructose already turned into the orange color, and also glucose. Blue color solutions turn to the mud green color, then orange color, then as you can see, it turns into the brick red color. By using this experiment, we can easily identify reducing and non-reducing sugars. You can see all the reducing sugar turn into the brick red color. Fructose, glucose, maltose, lactose turns into the brick red color. But sucrose didn't change the blue color. Thereby we can easily say sucrose is a non-reducing sugar. At the end of the practicals, you can see all the other 4 sugar solutions change their color from blue, mud green, orange, and then brick red precipitation. This is a precipitate. I think you can't see it clearly, but this is a precipitate. But sucrose didn't change its color. So by using this, we can easily identify reducing and non-reducing sugar. Okay, thank you.
Okay. Then was it clear for you? And then when we performing the experiment, we did boil when we preparing the failing solution. So we boiling to mix the two solutions properly, and you have observed that when we doing the experiment, fructose gave the positive results within a very short period of time. That's because fructose has the highest reducing power compared to other sugars that we used. Okay. So, I think now you might have a question that in Fehling's solution and in Fehling's this Fehling's test, there's a redox reaction. Okay, then what will reduce and what will oxidize? What do you know about that? That's a redox reaction because reducing sugar, and on the other hand, Fehling solution is a mild oxidizing agent. There's a reducing agent as well as an oxidizing agent. And what will reduce? That means fructose or the glucose or the maltose or the lactose, they are reducing agents. Okay, then what will reduce? I mean,
Copper.
yes, copper 2 plus. You know that in Fehling solution, the active component is the Cu2 plus of bitartrate of cuprate 2. Then it will reduce. Cu2 plus will reduce to Cu plus. And the oxidation will taken place for reducing sugars. Okay, you know that when there is a free aldehyde or ketone group, it can be further oxidized into carboxylic acids. Okay, that is the redox reaction. There's a reduction as well as oxidation. Okay, then can you see this? I just prepared this to give you an understanding about this. Fehlings A contain copper sulfate and Fehlings B contain Rochelle salt and sodium hydroxide, potassium sodium tartrate. This is the chemical structure for potassium sodium tartrate. And when we prepare in the Fehling solution, what we do is we mix the equal amount of Fehlings A and Fehlings B, and it will form this kind of a complex, and this is called a bitartrate of cuprate 2. Okay, then Cu2 plus is the active component here. Okay, then this is the redox reaction in the Fehling test. Cu2 plus will reduce into Cu plus. Okay, Cu2 plus is in the Fehling's solution, and that means oxidizing agent is the Cu2 plus. Then it will turn into Cu plus. And this is, what is this? I have drawn a chemical structure. What is that?
Fehling.
Sorry, what is this chemical structure? Yes, glucose. This is glucose. Okay, and glucose will oxidize into, see here you can see a free aldehyde group CHO, and it has oxidized into COOH, that means carboxylic group. Okay, then you know that in the Fehling solution it has a basic medium, so sodium hydroxide is there. You know that in Fehling's B it contains sodium hydroxide. Then in the presence of sodium hydroxide with Cu plus, it can form cuprous hydroxide. And in the Fehling test, we do heating. When we do heat, this cuprous hydroxide can be converted into cuprous oxide. Okay, and cuprous oxide is the brick red color precipitate. Okay, then this is a brick red color precipitate. Okay. Then why we do need this potassium sodium tartrate? Then definitely you might have a question. In Fehling solution, the active compound we need is Cu2 plus. It contained in the Fehling's A. Okay, in Fehling's B, then why we at Fehling's B, and why specifically this tartrate component should be there? Why is that? To keep the solution in alkaline medium, yes, that's the one objective. You know that in Fehling's B contains sodium hydroxide. Okay. One thing is to provide basic basic medium sodium hydroxide. And there's another thing. Why specifically we do require potassium sodium tartrate? Tartrate is not a basic thing. Why we need tartrate? Okay, tartrate is there to form that complex bis tartrate to cuprate 2 complex. Okay, we do require to have that complex because when there's especially alkaline medium hydroxide, the Cu2 plus can form cupric hydroxide. Okay, then when we do heating, this cupric hydroxide can convert into cupric oxide. Okay, then what is the color of cupric oxide? It's black. Cupric oxide. This is cupric oxide. When there's no tartrate, okay, when there's no tartrate, Cu2 plus can react with hydroxide and form cupric hydroxide. Okay, the number of copper is the redox number is 2 plus, and it can form cupric hydroxide. Okay, then if cupric hydroxide form in the solution, we cannot observe our results. Okay, therefore tartrate, presence of tartrate is very important. Okay, so the tartrate remove this reaction from the solution. Okay, then there's no free Cu2 plus to form cupric oxide, since there is a complex that will not react with hydroxide, bare hydroxide. Okay, then this is the reaction that we intended to have, Cu plus hydroxide, and cuprous hydroxide should be formed, and it will react when we do heating. It will turn into cuprous oxide, which is the Picric precipitate. Okay, is that clear? If I were a student, so I may have that question. That's why I draw this redox reaction and explain that to you. In Fehling solution, we do mix Fehling's A and Fehling's B. Okay, then that is the chemical reaction that is happening in the mixing, and we do heat to facilitate this chemical reaction. And in the Fehling solution, the active component is bis tartrate to cuprate 2. You know that copper is a D block element, and it can form complexes. Okay, this is such complex. Bis tartrate to cuprate 2. The active component is actually Cu2 plus. Okay, the redox state of copper is remained unchanged. Okay, then I explained previously that the importance of this tartrate, potassium sodium tartrate. Okay, so this is the redox reaction happening in the Fehling test. There's a oxidizing agent, that is the Fehling solution. Okay, and there's a reducing agent, that is the reducing sugar. Okay, you know that glucose, fructose, maltose, lactose, they are reducing sugars. In reducing sugars, they have free aldehyde group or free ketone group. Okay, then this free aldehyde group can be further oxidized, okay, into carboxylic group, and the Cu2 plus can reduce into Cu plus, Cu plus. Okay, and you know that in the Fehling solution, it contains a basic medium, so this Cu plus can react with sodium hydroxide and form copper hydroxide, cuprous hydroxide, not cuprous hydroxide, Cu1 hydroxide. And when we perform heating, this cuprous hydroxide will turn into cuprous oxide. This is the big red precipitate, cuprous oxide. Okay. Then I was explaining that the importance of potassium sodium tartrate. Okay. Potassium sodium tartrate can form complex with Cu plus. Okay. When there's excess Cu plus, you know that this happen in according to a stoichiometry. And when there's excess Cu plus here, it can react with sodium hydroxide and form cupric hydroxide. Okay, if that happens, when only Cu plus is there without that complex, this reaction can happen. Okay, then if this reaction will happen in the solution, that will form a black precipitate, that is cupric oxide. Okay, when that black precipitation is there, we cannot observe the cuprous oxide. Okay, that will interfere with the intended positive results of the test. Okay, that is why we have to avoid this reaction. Okay, we have to avoid that reaction. For that, we need to add potassium sodium tartrate. Okay, is that clear?
Yes, sir. Thank you.
Okay, so that's it, I think, for Fehling's test. And we will move on to Osazone test. Okay, this is very, very important test. You haven't done that in your advanced level. Okay, Osazone test. Have you heard about Osazone? Not ozone, Osazone. I think this is not in the, I think you haven't learned about that before. Okay. Ozone is a class of carbohydrate derivatives found in organic chemistry, formed when reducing sugars react with phenylhydrazine at melting point. Okay, this is a class of carbohydrates formed when reducing sugars or carbohydrates reacts with phenylhydrazine at melting point. It means 100 degrees of Celsius. Okay, phenylhydrazine. You know the chemical structure of phenylhydrazine, CH2, NH2. Okay, this phenylhydrazine can react with reducing sugars, particularly first and second carbons of the reducing sugars. Okay, then ozone test is very important because we can identify the type of reducing sugar using that test. Okay, in the Fehling test, can you distinguish between fructose and glucose? Fehling test again, fructose with glucose. Yes. If you are very vigilant, you can identify, because the reducing power of fructose is higher than that of glucose. But yes, if you are vigilant, you can identify, but that's not a very good way. Okay, then can you identify glucose and so say lactose and maltose using the Fehling test? Lactose and maltose. What is your opinion? Yes, we can't. Okay, then ozone test is useful in that sense. We can identify the type of reducing sugar. Okay, all reducing sugars react with phenylhydrazine to give products known as osazone. Osazones are a class of carbohydrates that forms when reacting that carbohydrates with phenylhydrazine at the melting point. Osazones are usually insoluble in water and used to differentiate reducing sugars because they show different characteristic patterns of crystals with different reducing sugars. In addition, different sugars convert to osazones at different rates. By noting the time required to produce the osazones and the shape of the crystals formed, the sugar can be identified. Okay, we use two figures. One is that actually three features to distinguish the type of to identify the type of reducing sugar. One is time taken to form osazones. Another thing is characteristic crystalline appearance and the melting point. Okay, we usually use the time taken to form crystals and the crystal shape. Okay, we usually not use that melting point thing. Okay, using that at a general laboratory, you can identify the type of reducing sugar. Okay, that is the importance of this test. Okay, you can identify the type of reducing sugar. Okay, if you go through the procedure, in here you have to take 5 milliliters of the sugar solution into a test tube. You have to add approximately 0.1 gram of phenylhydrazine hydrochloride, and approximately 0.3 grams of sodium acetate. This, please, as your next practical record, you have to write about ozone test. Okay, then in discussion, you have to write the importance of each step and why we add phenylhydrazine, why we add sodium acetate, and why we add glacial acetic acid like things. Therefore, please understand this clearly, and if you have any questions, don't be hesitated to ask. Okay. Then we warm the mixture until dissolved. We do warming to facilitate the reaction rate and to dissolve the content clearly, and filter the solution into a clean test tube and add a drop of glacial acetic acid. We filter to remove unwanted materials, and we add glacial acetic acid to provide acidic medium, and place the tube in a boiling water bath for about half an hour, and allow the tube to cool gradually in the room temperature. And we have to avoid rapid cooling. That is very important point in this experiment. We should not ever do the rapid cooling. We have to hold that spontaneously. Okay, if you do rapid cooling, the crystal formation will be disturbed, and you cannot observe the crystals. Okay, observe the changes, examine the shape of crystal form under the microscope and sketch it. Okay, usually in biochemistry practicals, we will provide you a super solution, and you have to do all these experiments. Okay, you are lucky. Is if you will not have in class examination, practical examination, you are lucky. But usually you have to do all these steps. This is a very key player in biochemistry practicals. You have to do all these steps. You have to very patiently do all the things, and finally observe that in microscope, and you have to identify the type of sugar. Okay, I don't know whether you will have exams in the online platform, or you have exams in here. I don't know, but this is very important test. Okay, so actually ozone reaction takes place at, as I mentioned, at melting point. It means 100 degrees Celsius at 4.3 pH medium. Okay, how do we provide an exact pH medium? We need some mechanism for that. What is meant by a buffer? Buffer එකක් කියලා කියනවා. In here actually we have a buffer. What is that? Yes, we use buffers to have a definite pH, or a pH to remain in a very narrow range. Okay, then here there's a buffer. What is that? We add only few sodium acetate. So can sodium acetate itself be a buffer? මතක් කරගන්න ඕගොල්ලෝ advanced level chemistry වල ඉගෙන ගත්ත නේද? weak acid එකයි ඒකේ conjugated base එකයි buffer එකක් වෙනවා කියලා. එතකොට අත්‍තර බලන්න මේකේ weak acid එක මොකද්ද? glacial acetic acid. And what is the conjugated base of that? Sodium acetate. Okay, in here we add glacial acetic acid, that is the weak acid, and its conjugated base, that is sodium acetate. Okay, there's other importance of adding sodium acetate is that it acts as a catalyst. Okay, sodium acetate acts as a catalyst as well as conjugated base to form a buffer. Okay, this reaction is taken place at 100 degrees Celsius at 4.3 pH medium. So in order to maintain 100 degrees Celsius, we do this test in the water bath. Okay, so for this approximately you may require about 40 minutes to complete this test. Okay, in biochemistry experiments, practical exam, final exam, you will have only 2 hours. Then you have to manage the time. You have to do this test, and you have to wait, place this in a water bath for about half an hour, and while that is in the water bath, you can do other experiments. Then you have to manage the time. Okay, likewise, this is not very easy course. You know that you have to develop skills, certain skills. Okay. Okay, then I will show you the practical procedure that we have followed. Then we will discuss that further.
Today we are going to practice Osazone test, which we are going to use for identification of carbohydrates. For that we have 5 different types of sugar solution: glucose, fructose, lactose, maltose and sucrose. First we have to measure 5 milliliters from each. Before that we have taken the sample into this beaker, and we are going to measure 5 milliliters from each of these solutions. First we are going to measure 5 milliliters of glucose. Likewise, we are going to measure 5 milliliters from each of these solutions. Then after measuring 5 milliliters, we are going to add them into separate test tubes. Now we are adding fructose. Then lactose. Then maltose. Finally, we are going to add 5 milliliters of sucrose into the test tube. Then after that, we are going to add 0.3 gram of sodium acetate and 0.1 gram of phenylhydrazine into each of these test tubes. We have added this sodium acetate and phenylhydrazine into the sugar solution. Likewise, we are going to add these two into our measured sugar solution. Then we are going to place these prepared solution in a water bath until they get dissolved. Okay? Then after the solution gets properly dissolved like this, when we see a clear solution, we are going to take it out of the water bath and we are going to filter these sugar solutions. After that, we are going to filter these solutions into another test tube. I think you can see it, okay? Then likewise, we are going to filter each of the sugar solutions we use today. Now you can see we have kept the samples for filtration, and after filtration, we are going to take this filtrate and we are going to, this is the filtrate, and we are going to add drops of glacial acetic acid into these samples. After addition of glacial acetic acid, we are going to place these samples in the water bath for about half an hour. We are going to practice this procedure for all of these samples. Now we have kept our samples in the water bath for about 30 minutes, and you may see crystals are formed in these test tubes, and it's better if you could notice the time taken for each solution to make the crystals. Now we are going to take these test tubes out of the water bath. I think you could see that some crystals have been formed. See here. You could see some crystals have been formed. After taking these test tubes out of the water bath, we are going to let them cool. We do not going to cool it rapidly. We are going to let them cool. After the samples get cold, we are going to take some crystals out of these test tubes and we are going to observe them under the microscope. First, we are going to take a slide and we take a glass rod and we are going to take some crystals formed, and we are going to place it over the glass slide, and after that we are going to place the slide on the microscope, and we have to focus it, and through the microscope we are going to observe different structures or different crystals formed by different sugar solutions.
Okay, then was that clear? Was that clear for you? So in the ozone test, then this is the ozone formation reaction. This is for phenylhydrazine. There's a phenol, the benzene group NH2. Yeah, that's it. This is called phenylhydrazine. And then you know that in reducing sugars, there's a free aldehyde ketone group, and usually this ozone formation occurs with first and the second carbon in a reduced sugar. Okay, only with the first and second carbon. Okay, then as the first step, one phenylhydrazine will bind, and another phenylhydrazine will bind here. Okay, then this is the osazone. Okay, then if this sugar is glucose, this will name as a glucosazone. Okay, then if this sugar is fructose, then this will call as fructosazone. If this glucose is if this sugar is lactose, the resulting ozone will call as lactosazone. If this is lactose, the resulting ozone will call as galactosazone. Okay, here you can see glucosazone. This is glucose. Okay. Okay, then we identify the type of sugar by microscopic crystalline structures as well as the time taken to form crystals. Okay, those two figures are very, very useful. First one is we allow that, you know, that after keeping that in the water bath, we get that and we allow to form crystals and cool spontaneously. And in the video also you have observed that fructose formed crystals at the very beginning. Okay, because you know that fructose has a higher reducing power, thereby it will form ozone crystals within a short period of time. Okay, then you know that what is the difference between glucose and fructose? Very, very, very tiny difference is there. Okay, in this is very tricky experiment. Even in the exam, we evaluate that glucose and fructose will give the same ozone crystalline structures in the ozone test, but we can identify we can distinguish between fructose and glucose. Okay, this is very tricky. Okay, therefore understand that very clearly. The crystalline structures of glucose and fructose are very, very similar. Okay, we can distinguish using the microscopic structures. Okay, here you can see, yes, monosaccharides like glucose and fructose will give this kind of needle-shaped appearance. Okay, and lactose will give this kind of powdery clusters, tight balls of needles-like crystals. And disaccharides like maltose will give that kind of sunflower or star-shaped crystals. Okay, I'll show you the figures that we obtained in the experiment. See, you will observe needle-shaped crystals in glucose and fructose. In the observations are much similar to glucose and fructose. Okay, and you will observe star-shaped crystals in maltose. Also, this is sometimes called as sunflower. Okay, then powdery clusters, this kind of powdery balls appearance will observe in lactose. Okay, you have to identify the type of sugar based on these observations. Therefore, please be carefully understand that. Okay, in the ozone test, we can distinguish glucose and fructose based on the time taken to form the crystals. Okay, what will form these crystals firstly? We have observed that, as well as I mentioned that a few minutes back. What will form glucose this crystalline structures or osazones first? Will it be glucose or fructose?
Fructose.
Fructose, yes. Then reducing power of fructose is higher than that of glucose. See, this is glucose and this is fructose. Then see the difference between glucose and fructose is that glucose has a secondary hydroxyl group while fructose has a primary hydroxyl group. Okay, in first carbon there's a hydroxyl group or OH group. In glucose, in the second carbon it has a hydroxyl group. Usually this primary hydroxyl group is much reactive than the secondary hydroxyl group. Okay, therefore reducing power of fructose is higher than that of glucose. Okay, since the phenylhydrazine react with only the first and second carbon of the sugars, the final product will be same for fructose and glucose. Therefore, even though we use of fructose and or glucose, you will observe needle shape appearance. Okay, the final product is same, but the time taken to form crystals is different. Fructose will produce the final product within a short period of time, usually within less than 2 minutes. But for glucose, it will take about 5 minutes. Okay, see. මේ ග්ලූකෝස් වල ෆ්‍රක්ටෝස් වල වෙනස තියෙන ඇත්තටම ග්ලූකෝස් වල OH group එක තියෙන්නේ දෙවෙනියට ෆ්‍රක්ටෝස් වල තියෙන OH group එක පළවෙනි කාබන් එකේ. එතකොට මේ පළවෙනි කාබන් එකේ තියෙන OH group එකක් තියෙන OH group එකක් තියෙන ඉඳ මේක ග්ලූකෝස් වලට වඩා රියැක්ටිව්. හරිද? එතකොට reducing power එක වැඩි කියලා කියනවා. එතකොට phenyl hydrazine react කරන්නේ පළවෙනි සහ දෙවෙනි කාබන් එකත් එක්ක විතරයි. එතකොට අන්තිමට හැදෙන product එක ග්ලූකෝස් උනත් ෆ්‍රක්ටෝස් උනත් සමානයි. නමුත් මේ අන්තිම ප්‍රොඩක්ට් එක හැදෙන්න යන කාලය මත අපිට මේ දෙක අයිඩෙන්ටිෆයි කරන්න පුළුවන්. ෆ්‍රක්ටෝස් වලට විශේෂයෙන්ම යන විනාඩි දෙකකට වඩා අඩු කාලයක්. නමුත් ග්ලූකෝස් වල විනාඩි 5ක් විතර ප්‍රමාණයක් යනවා. Okay. Usually in the exams we evaluate these things and you have to distinguish glucose and fructose based on your observations. Okay. Therefore, understand that. Okay. So yes, and these are the observations that we usually see under microscope. Glucose and fructose, you will see this type of needle-shaped appearance, and star-shaped appearance will be there for maltose, and powdery clusters appearance will observe for lactose. Okay, these crystals have definitive structure. Okay, based on that you can identify the reducing sugar. Okay, will it give positive result for sucrose? Will it give this kind of crystal shapes for sucrose? No, because sucrose is an unreducing sugar. Therefore, there's no free aldehyde or ketone groups to react with phenylhydrazine. Therefore, non-reducing sugars will not give positive results, or crystal or ozone formation will not happen for non-reducing sugars like sucrose. Okay, the ozone test is important because we can identify the type of reducing sugar using this test. Okay, or what could be the reason for that? What could be the reason for that? Only using the Fehling test, we could only observe, we could only distinguish between reducing and non-reducing sugars, but in this test, you can identify the type of reducing sugar. Okay, is that clear? This test is sometimes in some textbooks I have seen that this test is referred as a phenylhydrazine test. Yes, phenylhydrazine test. But it's better to refer that as ozone test. Okay. Is that clear?
Yes, sir.
Okay, then you have to write a practical record based on the ozone test and write observations and include figures. And I can show you the yes. Can you see this image? Yes, these are observations that we got. So okay, and also if you need any further details, I will. Okay, I have, and also uh, I forgot to tell you that we have used five sugars in ozone test: fructose, glucose, lactose, and maltose. And fructose will give the results within less than 2 minutes, and glucose will give results uh, within about 5 minutes. And that's tricky. You know that the crystalline structure is similar, and you have to identify based on the time. Okay. After that, lactose will give results after about 25 to 30 minutes, and maltose it will get about 45 minutes. So if you get maltose in the exam, so you will be sometimes in trouble. Okay, but we give you that. It's not at the actually it's not a big deal. Just you have to wait, patient. You can manage other things. Okay. So just remember the crystalline structures. Glucose and fructose, that's needle shape, and lactose, it's powdery cluster shape.
Excuse me, sir.
Yes, yes,
When we are writing the reports,
yeah.
do we need to draw the instruments that we use?
Uh, no, no, it's not necessary. Instrument means what?
That we use in the practical.
Uh, for an example, what kind of things?
That means that then we use the Bunsen burner for the burn. So do we need to draw that when
Uh,
we?
if that in under methodology, if you wish, you can just uh, try the set out. I mean, draw the instrumentation. That's okay, but I thought that when you are typing the things, it would quite difficult. But if you can, that's good. Okay, that even that will helpful to remind you the things. That's okay. Maltose is touching. So this is your practical report number 2. You have to do that for ozone test. Okay. Do you have any questions? pH level of the practical. Which practical? pH level means what? I didn't get that. pH level of this practical. In ozone last practical, it means ozone test. You have to maintain pH. Usually, this ozone formation oh yes, this is pH dependent ozone formation, and as I said, ozone formation happen at the melting point. It means 100 degrees Celsius melting point of water. Sorry, at 100 degrees Celsius at 4.3 pH. Okay, so we can maintain the 100 degrees Celsius, not melting point. It's not a melting point. 100 degrees Celsius, we can assure that there is 100 degrees Celsius when we use a boiling water bath. Okay, and we have to maintain 4.3 pH. Usually, when we use this buffer, sodium acetate and acetic acid buffer, we can say that we have that pH. We usually don't check the pH, but this reaction is occurred at that specific conditions. 4.3 pH and 100 degrees Celsius. Okay, that is why we add glacial acetic acid and sodium acetate, as well as doing this test in boiling water bath. Can we add some photographs regarding the experiment under method? Yes, you can add. You are free to do that. Okay. Usually, practical record is, I don't know if you can see this. No, I think this is not clear. No. See, that one student had asked that it's okay to add right raw figures. Yes, it's better if you can add right. I mean, draw the figures. That's okay. And chemical structures. And now under observations, you have to draw the structures of the osazone. Yes, even this is for osazone. Yes, and I forgot to tell you that when you are writing the practical records, it's better to write objectives. Okay, there's a specific objective for each experiment. For an example, for Fehling test, objective is to distinguish between reducing sugars and non-reducing sugars. Okay, then write that in the under objectives. So as a sequence, you have to write date, title, practical, date, practical number, title. Yes, then after that, write about objectives. That's better. Then introduction, methodology, yes, observations, results, and calculations, and after that, discussion or conclusion after discussion, and finally references, and also include references. Okay, so yes, objective is different from one test to one. Okay, then as I mentioned, Fehling test is to distinguish between reducing sugars and non-reducing sugars, but in ozone test, it is to identify the type of reducing sugar. Okay, we can distinguish reducing sugars and non-reducing sugars also, but the primary objective is to identify the type of non-reducing sugar. Sorry, reducing sugar. Okay. Likewise, objective also has to be included in the practical records. Okay, I'll usually we give only 3 days to submit the practicals, but since this is the first week, I gave you about you have to submit the first practical record in Monday, as well as this in next Wednesday, I think. Yes. Okay, I'll take only 5 practical records. Okay. Other than that, do you have any questions? Don't think this as a burden. Just actively participate to this. Yes, what would be the page limit for this practical report? So this is a bit lengthier than the previous one. Actually, you don't need to write about the Fehling test and the MOS test. I just wanted to include about ozone test. Okay, thereby usually I don't expect a very lengthy assignments or practical reports from you. Just 5 pages. 5 pages, including the front page, is enough. Okay, you have to manage that. Okay, quantity doesn't matter, but the quality. Okay, you should be able to summarize the things and manage the limit and the time you have. That is very important, very important thing. Okay, thereby just limit it to 5 pages. Okay, so less than 5 pages is most welcome. That's fine. Okay. Okay. Other questions? Okay, it's up to you. You can utilize me effectively, or you can just hear this and forget the things. Okay, but I don't know. I have worked hard for this practicals. I could just prepare a video or take photographs and do you this as a presentation and finish the things. But I think this must be effective. That's why I record the things and edited those things for you and doing this for you. Thereby, it's up to you to how you utilize me. Okay, that's okay. I'm not blaming anyone. Okay. Then if you do not have further questions, I will stop the lesson from this, and I will upload the necessary things to the LMS. And thank you for your kind participation. Thanks a lot.
Thank you, sir.
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 Thank you.
Thank you, sir.
Thank you, sir.